Well, welcome everyone. Thank you for joining us tonight. My name is David Dodick. I'm the chair of the American Brain Foundation. And, we do these salons to bring some of the most renowned neuroscientists into your living room and to raise awareness of brain disease, provide a state of the art on where we are with regard to a particular topic or disease, highlight the importance of research in getting us to life-changing treatments and cures, and also to thank you for your generosity, having given generously to the American Brain Foundation to help us support research. To that end, we've supported almost 300 incredible young scientists with over $40 million in research money over the years. And the vast majority of those people, we're very proud to say, have gone on to have prolific careers and are making tremendous contributions. But as you know, research is costly, and it's going to take a sustained and even more tenacious effort to get us to a point where we have treatments for brain diseases that, according to the World Health Organization, represent the leading cause of disability in the world and the second leading cause of death in the world. So tonight, we're going to hear a presentation entitled Cure One, Cure Many: Connections Between Brain Diseases. And that's really what forms the basis of our vision and drives our research. We fund research across the entire spectrum of brain diseases, not just single diseases, because we believe there are common threads in terms of disease mechanisms that link them, such that if we find a cure for one, it will have a ripple effect and indeed may represent a cure for many. So let's get into our topic of conversation for tonight with, and I'm very honored to have our guest speaker here, Dr. Frances Jensen here. She is a professor of neurology and chairman of neurology at the Perelman School of Medicine at the University of Pennsylvania. She's the co-director of the Penn Translational Neuroscience Center. She was formerly a professor of neurology at Harvard Medical School and director of translational neuroscience at Boston Children's Hospital and Brigham and Women's. She's the current president of the American Neurological Association, and we're proud, and I'm proud to have her as a director on the board of the American Brain Foundation. So, Frances, thank you so much for being here tonight. The floor is yours.
Well, thank you very much. And thank you, David. Thank you, Jane Ransom, for inviting me. And of course, I am very proud to be a member of the ABF board. There is something really special going on here, and I hope I get a chance to convince you tonight. So let me try to share my screen. And I think you are now probably seeing a version of my, is everybody seeing my slide presentation and not my email?
Yes.
Good. Okay. All right. So I'm going to talk to you a little bit about the impact and opportunities in brain research, especially as it pertains to neurology and the clinical neurosciences, and why this is such a absolutely critical moment in our field. This new neuroscience, clinical neuroscience, new neurology is truly the fastest moving area of biomedicine and clinical science, bar none. And it's built upon a foundation of research advances that have been happening in the last, 15 to 20 years, as well as a lot of incredible new high-tech clinical diagnostic methods. So we have now a really sophisticated technology that is moving closer and closer to the patient. The basic research is absolutely necessary in animal models, cell cultures, organoids. But what you're seeing in this last 10 years is a move to perform actually basic science in the patient, in human biosamples, on human people, because of the diagnostic technology that we can apply clinically. And they're increasingly acknowledged, now as some of the most prevalent disease sets, as David mentioned. So actually, when you look at this, and this is actually a most recent version of this graph, we look at this thing called the disability adjusted life years, which is like the basic, how many life years have become disabled by a disease set, essentially. What's the burden of the disease? And this is a IHME 2021, I think, 2020 publication up to 2019 about the diseases and their relative weight in terms of how prevalent and how problematic are they. Initially you go, well, I don't see the brain up at the top here. Well, that's not entirely true. If you look at, you could even include musculoskeletal disorders, but substance, mental health, neurological disorders, and self-harm, which of course comes in psychiatric disorders, we're really talking about 20% of all disability adjusted life years, which actually overtakes any of these other numbers here as we cluster. And you know what? This actual slide is an example of the problem that we have talking about brain diseases. It seems fractured. The public kind of label things differently, but we know this is all brain. And the brain is, composed of a myriad of component parts that are sort of reorganized, rearranged in space and time to create different diseases. And that's where the cure one, cure many sort of mantra really hits home. So these are all very related, even though the IHME and many people in society consider these all very different. And in fact, when you look at the burden of neurological diseases in the United States, you can see that the cost of these diseases to our society is enormous and growing, especially age-related neurodegenerative disorders, which are, of course, on the rise and an alarming rate. I want to tell you, though, that we've had an incredible last 7 to 10 years in our field where things have happened in these disorders that are frankly unimaginable to me, would have been unimaginable to me when I started my career 30 plus years ago. We are reversing strokes with TPA or TNCase, and we do thrombectomy up to, meaning taking the clot out of the vessel up to 24 hours after stroke. It never was used to be that somebody had a stroke and they would potentially be disabled for the rest of their life. We're seeing people go back to work after these procedures and go back to their normal life. Epilepsy has seen 30 plus new anti-epileptic drugs in the last couple of decades. We're now using lasers and closed loop stimulators to modulate brain activity. I'll show you a little bit of that in a minute. Multiple sclerosis, another extremely common disease that affects the lifespan. We have immunotherapies that are game changers, something called Ocrevus, ocrelizumab, and many second generation drugs are being used now that are really decreasing the progression and the burden of this disorder. Movement disorders, we have deep brain stimulation, focused ultrasound being used on people's brains through using a lot of computational analysis of brain activity and networks to help regulate brain network loops that are involved in Parkinson's, in Huntington's. We have new biomarkers and other great drugs for these because we're understanding the basic biology of these disorders. Cognitive neurology, of course, similarly, these age-related disorders, we are now being able to much better predict dementia. You all know about aducanumab, Aduhelm. It's not going to be the blockbuster I think it had been hoped to be, but it's a beginning of a path down a really amazing road that is really a result of our better understanding of the kinds of proteins that get accumulated that trigger this neurodegeneration. And we do believe that that is like a very exciting new area that is going to really probably yield in the next decade some great results for treatments. Another huge game changer is the ability to do gene therapy on patients and actually inject through the blood or through the spinal fluid or even into the brain genes that will change disorders like that used to be due to mutations, genetic mutations. And this is really an amazing area in brain science right now. Ophthalmology, we have new ways to look at retinal disease. And it turns out retinal disease, you know how they say the eye is the window into the brain? Well, it turns out that there's a lot we can understand by watching eyes to tell us about some of these neurodegenerative disorders. There's things like autoimmune panels, looking at immune antibodies that are telling us about diseases that we thought were just what we called idiopathic diseases. We didn't really know what they were, but they're things like amyloidosis, which causes peripheral neuropathy, heart problems, many other issues, autoimmune encephalitis that I'll tell you about in a minute. We have even new drugs for migraine, as David knows, these CGRP inhibitors. These weren't even around 5 years ago, and this comes out of the research that's been exploding in neuroscience. And then finally, neurocritical care is a great example where state-of-the-art neuroinformatics, AI, computational analysis, they're getting terabytes of data from patients in the ICU and beginning to put it together to see through neuroinformatics, through AI, beginning to see patterns that the human eye can't see by itself. I hope that you just appreciate what our field has accomplished in the last 10 years, or 7 years, actually. It's extraordinary. We can only hope for more, and I truly believe we're at an accelerative moment. So one of the things that really resonates for me, and has always resonated for me, is this idea of cure one, cure many. All of those examples of cures that I just showed you are not isolated researchers working on one single disease at a time. They're sharing information. Something somebody might learn in migraine might inform epilepsy. Something somebody might learn in dementia might inform Parkinson's disease. And I think we are beginning to see this kind of acceleration because people are communicating, and actually scientists are sharing information, and they're not only focusing on a single disease. And when I think about brain diseases, if you think about this as sort of like an example, there are many, many more silos I could put here. It used to be that we used to think that epilepsy was a disease that was kind of unto itself. Nobody would have, it was just epileptologists that could work on this, and that there was nothing from multiple sclerosis over here, MS, that had anything to do with epilepsy. Well, that's not true anymore. We're realizing as we break down mechanism and we look at human tissue and human imaging, genomics, genotype, phenotype, we're seeing that these are actually way more related than we ever thought. And here's an example. These crosshatches are examples for you of things that absolutely bind together different disorders. And actually, we can share information from one to the other. And in fact, we're trying with, even at the industry level with clinicaltrials.gov, we're trying to get results, all results of clinical trials, for instance, out, available for people to share, because, a negative result that really nobody cared about the epilepsy, say in some dementia patients, and there's a big study going on, it turns out that maybe something people learn from a study of a protein or gene in dementia actually syncs up nicely with like very rare observations in epilepsy that could actually show us a new connection between the two diseases. And so we're hoping that more data mining, more sharing of mechanism will actually create a better field. It's almost like we should change the nosology of our field. These are old, archaic terms for diseases that actually can be described more as channelopathies or disorders of metabolism or protein accumulation disorders. And inflammation, of course, we're learning goes across all diseases. And we see the same players rearranging themselves that are seen in migraine, for instance, into MS. And from MS, they rearrange themselves, and we can see some of the same players in epilepsy, TBI, dementia, and so on. So we need to be sharing information. And that is what this message about cure one, cure many is about, that you set talented investigators to focus on a single disease. If you don't ask them to look outside your disease, their disease, we're missing a lot of opportunity. And in fact, clinically, we're also able to use the very same on patients. We're able to use the very same metrics, and we can gather data. It's very similar across different types of diseases, and go mine the data for similarities. So not only the cellular and molecular connections are there, but some of the same kind of data is being gathered from these different patient sets. So neuroscience, has advanced unbelievably. We have genetic screening, imaging, brain connectomics. We're learning about regulation of genes. Epigenetics is a huge new field that it crosscuts every single disease. The use of stem cells, immunology, as I've talked about, brain inflammation, degeneration, and also thinking about interfaces between brain and machine. And if we think about like imaging, how important imaging is, we can actually evaluate using DTI methodology and functional imaging, we can show parts of the brain that are connected to one another. We're very, very dependent upon imaging to understand the complexity of our brain. And actually, we can take those pathways that I just showed you in this field where, you're looking at what is this pathway, might be touching, moving from the thalamus to the cortex or from one side of the brain to the other. We can track these tracks. So we're tracking tracks and we're sort of understanding the wiring diagram of the brain. We can reduce that information to like models like this, where, well, that we know is attached to this part of the brain. Is it involved in a disease? And when it connects to that part of the disease, to the brain, maybe there's a seizure, for instance. So we want to understand the wiring diagram of the brain and how important it is in helping us with understanding diseases. And we know that we have science-based understanding now of this wiring diagram and how it's affecting many, many different areas. Again, understanding this opens the door, unlocks the understanding of a lot of different network disorders. An example, really obvious example, is in epilepsy. This is actually done now. This is applying basic science to human brains and taking engineering concepts. I love this slide because so much is going on here. This is one of those, way we analyze pathways where you can track tracks in the brain with high resolution MRI. So all of these different colors are just showing that the red are all connected to a deep area below that is red. We pseudo colored it, but it's giving us like a map. And they can reduce that down to something where they can look at different areas of the brain, for instance, and now reduce it down to some more basic wiring diagrams. Meanwhile, in the human epilepsy patient, for instance, we can place electrodes around the outside of the brain and see how a seizure might propagate across one of these networks. It can actually, through advanced imaging now and artificial intelligence, essentially, we're able to sort of track the seizure and find out where is it coming from and where does it go to, and where would we ablate to stop that spread to allow the brain to function normally again. This is the exact kind of work that is being done that has taken years now to now be in place, and we can use laser therapy to actually ablate, say, the source of the seizure based on this kind of science. Circuit disorders are not just epilepsy, Parkinson's, tremor, other, reactivating, constantly in motion activated neuron networks are other examples. And for sometimes we can use programmable interactive electrodes that will sense that area that we've identified as the problem and either activate it or deactivate it to cause a cessation of an abnormal tremor or a seizure, for instance. So this is the kind of futuristic work that is happening now. I mean, it's happened, it's now, and it's being perfected with even more advancements. This is where we're at now. This is my reality as a neurology chair. This is what we're actually offering patients. We have non-invasive means of regulating activity. This is another cross-cutting area. We're trying to figure out how do we change that wiring diagram to stop one part of the brain activating another. We can go in with stimulators like I've just shown you, or we can use external stimulation from, say, transcranial magnetic stimulation. This is based on years of biomedical engineering work around magnetic fields, the same kind of magnetic fields we get MRI imaging from, which will actually stimulate brain. And if you turn on or off, if you will, an area of brain that is either underactive or overactive, because we've used that wiring diagram to understand its importance in a symptom set. Right now, it's mostly in neurological disorders, but it's also being used in depression and soon to be used in other psychiatric disorders as well. And this is where these wire coils is actually using magnetic fields to activate these little neurons in the brain to turn on or off different areas of the brain. And in fact, we can pair it with physical or medical therapy when somebody has a stroke, for instance, because we can map areas of the brain that are too active or not active enough. We can, from the outside of the brain, actually stimulate them to help somebody who's trying to learn to speak again or trying to learn to use their arm again to activate a area of the brain that might be nearby an area of a stroke or brain injury, to sort of boost physical activity, for instance. And there's a lot of neuronal activity that we understand around that. What about brain injury? We're learning a lot about brain injury. Post-TBI, we're able to model this in animals, but we're also able to look in humans now, taking blood samples at days or hours after their injury and pairing it with advanced imaging and also blood tests and blood samples. And we can see different proteins that are present at different time points after, say, TBI. And it might be one day that we can find that, one kind of signal is telling us that this person is going to have a very rough course and will need new therapies, and others, no, these people are, this person is going to have a mild case. So we have the ability to do this. I have somebody drawing on my screen for some reason. And I don't know how that happens, but anyway. So the next thing I want to talk to you about is the role of AI and overlaying a massive amount of information that we have on with respect to genomics and being able to understand people's genes and this ability to see brain activity and functional MRI imaging. So I showed you that wiring diagram a minute ago, but you can see that we've come way beyond that. You can get many, many points. And what we're able to do now is actually take that with advanced with artificial intelligence, frankly, being able to do computational analyses of what's on or off and match it again against a genetic set and say, oh, this is the pattern for schizophrenia. This is the pattern for epilepsy, and actually begin to use these maps independently to corroborate what used to just be a behavioral observation. We're really starting to get to the nature of being able to sort of identify in a much more objective way some of these softer signs of brain dysfunction. We also are, making headway with respect to neurodegeneration. As you know, many types of dementia, 1 in 10 people over 65, 1 in 2 over 90. It's 4 times the cost of care, with somebody with dementia with not. It's a huge problem. And right now, where this field is, it's more at the point of looking for markers of who's getting the disease. So we can then take that patient group and try new therapies on a more pure group. And you can see here, we have amazing tools to look at these pathologic proteins. So in many cases, what we have are pathologic proteins that are building up. And in the case of, we can do PET scans to find these proteins. We can overlay them on high resolution images to see exactly the brain cells that are involved and take that back to the lab, to animal models. We can use antibodies, which is what aducanumab was, which is what these things are, where you can see like they're trying to absorb to take away these abnormal proteins, and you can see that. And that's where these antibodies, there's anti-tau, anti-amyloid, anti-synuclein. Tau and amyloid are mostly seen in dementias, and synuclein in Parkinson's, and a combination can be seen in other combinatorial diseases and Lewy body disorder, et cetera. But you can see these are different diseases, but the same idea that you need to use an antibody to take away an abnormal protein. So again, this kind of science has come out of the cure one, cure many sort of mindset. Neuroinflammation, the most common form of neuroinflammation that we all think about is multiple sclerosis. It is, we know it is an inflammatory disorder of different parts of the brain and spinal cord. It can attack anybody at any age group at any time. And what's actually happening are you are developing antibodies against a very special part of your brain cell called myelin, and you are losing myelin largely, among other things, and it results in an inability to conduct signals or send messages from one side of the brain to the other, or one part of the brain to the other. And we can see this now on imaging, where it's sort of those tracks that I showed you, those beautiful colors, are kind of involved in this under the surface of the cortex here. And you can see the white means that they've been damaged. This would be an example of somebody with multiple sclerosis. And multiple means that most people have multiple areas of sclerosis, which is scarring, essentially. So we have many different methods now of understanding like how to get at this. There's new antibodies. There's looking at the role of different immune cells in the brain. New targets are being developed. And when I told you earlier, Ocrevus or ocrelizumab and that class of medications comes from research on these tiny lesions and understanding what tiny cells are doing. And here we have inflammatory cells that are inflammatory cells that are attacking the myelin. So here's the myelin sheath. Here's a little neuron. This is a myelin sheath. And the oligodendrocyte is a little cell that wraps the myelin, which is this nice coating to allow signals. It's like the insulation on an electrical wire to make a signal go fast from, say, this neuron out to the next neuron. And these little oligodendrocytes are under attack by cells called microglia and other inflammatory cells. And we understand a lot about these cells. Now, what we're understanding, though, is that there are, it's not just these cells, there's other mechanisms of inflammation that are being activated as well. And turns out we have many, many targets that can be approached. We're also learning, though, that these microglia can be very good for brains. And here is for our brain health. Here in a normal, healthy brain, here's a brain cell, and it's sending signals out across a synapse from this white cell to the red cell. And the microglia are helping keep that synapse healthy. But when they do something bad, they get overactivated and they can also attack the synapses. So not only can these microglia, when we need them, we can't get rid of them, but when they overact, they can either attack the myelin and the oligodendrocytes that are making myelin, or they can actually attack the brain cells themselves. And when that happens, because we're studying now microglia, not specific diseases, we learn that other disease types can happen. And one example is something called autoimmune encephalitis, which is parts of the brain can be attacked by microglia and other cells and antibodies against brain cells, and create a inflammatory process that can result in, initially, a person may be feel bad, they may have psychosis, then they might start to get headaches, then they might start to get seizures, and then they progressively deteriorate into a coma. And 25 years ago, we didn't know what that was. They couldn't find a virus, you couldn't find an infectious agent. And then only recently it was understood that it's an autoimmune attack of the brain on itself. And an example here is a brain cell, which is the bright green are the antibodies coming from these inflammatory cells that are basically draping themselves over the entire brain cell. And no wonder you're getting these, a brain-wide, what we call encephalitis. But we now have antibodies. Now that we know what the antibodies are, we have treatments specific to these different forms of limbic encephalitis based on the science. And that is now being used routinely in medical centers. So what used to be a devastating disease, and you can see it's a pretty severe thing going on there, we have treatments for because of this sort of connected science that was done. So I hope I've given you some examples of how it's a reality. This is not a pipe dream anymore. And I think what the thing I find when we talk about brain research, often we're always talking about, well, one day in the future, we'll do this, we'll do that. I'm here to tell you that it happened. We have advances already from this last decade even of incredible cross-cutting brain research because of this way of thinking about breaking down silos. And what we have right now is this cycle that you all have heard a lot about, where you start off with the early discovery work being done at the bench. You move forward into the first translational into humans. That's called T1. T2 is actually, that's like a lot of my job as a department chair, getting it out for the first time into clinical settings. And then it becomes de rigueur, and you have to look at populations to see how they responded. And then you will find new questions that come out of populations that you bring back into a question for somebody to ask in a brain cell, preparation or a lab, and then the cycle begins again. And I would say that the American Brain Foundation is really here to support this cycle. And what do we need? We have pre-federal funding for startups. We can't, a lot of what I've talked about, when it first started, it was not funded by the NIH or anybody for that matter. And often because these are interdisciplinary, because they don't fit into a silo of a specific disease group, it can be hard to get these funded. And I think this is what we need. This is like 21st century research funding. It's not like the old way of doing things. It doesn't fit traditional funding mechanisms, what I think we are talking about here. And as you can see, we also need to bring in other fields. I've just shown you, I hope, so you can appreciate how important bioengineering is. And we need to manage big data. There's a lot of AI and computational work to be done in our field. And we need to stratify patients. And we also need to pay attention to rare diseases, because occasionally, or not occasionally, a rare disease will highlight a specific pathway that, like a molecular pathway, that actually is very, very major in that rare disease. But actually, we realize, because we do this cross-cutting work, that it's involved in many forms of diseases. It's just not the biggest player, but can be a really important therapeutic tag. So to close with, I think, I hope I've given you a sense of, like, this is not a field that's in its nascency and just starting off. This is a field that is revved and midstream, but needs more priming of the pump, because it's going somewhere and it's changing so rapidly. And I think ABF has the unique attribute of not being a single disease centric funding organization. So I think it has the opportunity to take advantage of discoveries that are emerging in one field and help us expand and amplify the importance of those findings. So I hope you have appreciated how fast our field is going, and I'm happy to take questions. Which may be in the chat. And I can't hear you. Jane, you're muted.
There we go. I got unmuted. They muted me again. Frances, that was a brilliant presentation. I thought I was excited about this. You got me even more excited, and I'm sure you got the audience excited too. Just where we are in terms of state of the art and how close we are to finding really life-changing treatments for many of these diseases that we deal with. We've got a big audience here with a lot of questions, but I'd like to ask you a question to start off with, and that is that you touched on the need to break down silos within disciplines of neurology. But I also think we need to break down silos, and you talked about bioengineering too, so areas outside of medicine. But I think we need to break down silos across disciplines of medicine, because many of the most important risk factors for neurodegenerative disease, stroke, the diseases that are most disabling, as you outlined, are things which we don't do a good job of managing, like hypertension. We've got 120 million people in the United States with hypertension, and about 1 in 7 are actually well managed. Obesity, we got 90 million people with obesity, and obviously not well managed. So when we just think about the modifiable risk factors out there, the Lancet Commission says that 40% of dementias and 60 to 80% of strokes are preventable. So we really have to work well, I think you'll agree, with other disciplines and other specialties to really just get the fundamentals right.
Well, yeah, I mean, I would say that, the cycle I was just showing you with the T4 being the population, actually, those associations that you've just mentioned hadn't been known until we did population-based research to go, oh my God, actually, it's not all Alzheimer's. In fact, the most common cause of Alzheimer's is vascular dementia with Alzheimer's. And the most common cause of dementia, sorry, is vascular dementia with Alzheimer's. Population research is, now that we have the computational wherewithal to do these large scale studies from all these different databases, is showing these connections that you're absolutely correct, that between diabetes, hypertension, cancers, different cancers that can interact. And it also is shedding light on the sort of public health disparities, certain populations that are more prone to something. There are hidden messages in all of those things that you can reduce down to a basic molecular pathway, if you wish, to look for new therapeutic targets and better understanding or prevention of these diseases. We are in an extraordinary moment in medicine in general. I agree with you. And this applies to the neurosciences. The neurosciences have sort of been a little bit in the back because people are like, oh, we don't know what's going on with the brain. It's a big black box. Not anymore. And like, we're in. Another thing that you mentioned that I think is something that people should think about, and I do think it it's another sort of cure one, cure many kind of topic, is lifespan research. And the fact that, just like people know the whole story with diabetes, right? Obesity in childhood increases your risk of diabetes as an adult. But there are disorders of the brain that, when affected early in life, can affect your risk of dementia later in life, or epilepsy for that matter. So we're understanding, that things are a lot more complex. And that's where this cycle really is important, because you have to be informed by the human condition as well, and bring that population observation back in to say, test it as a hypothesis. What is that? Why is that the case?
Yeah. There's a couple of questions here about phototherapy and stem cell transplants for Parkinson's disease. Do you want to touch on any of that?
Yeah. So, mentioned that Parkinson's disease, for instance, is we know its effects. It comes from generally a focal area of the brain. It's certain neuronal populations that are lacking dopamine, but we also know that it's way more than that. That those populations are sending messages through those pathways that I showed you to other parts of the brain, and are being influenced by, similarly influenced by parts of the brain that are acting on those populations. So we understand it's sort of a network disorder. So we want to make sure that's why the DBS is working quite well in people. You can either supplement the dopamine with many different kinds of drugs now, but sometimes that isn't enough. And the DBS, where you can actually directly stimulate or turn on or off different parts of this network, has become increasingly used in people that don't respond well to drugs. Now, researchers have been actively looking for, can we put in stem cells that can go and replace the cells that are depleted of dopamine and are degenerating? Can we replace those? So a lot of research is going on about what is it about the cells that you put in? Will the brain tolerate it? You won't have an inflammatory reaction? Will they make synapses to the other parts of the brain that are important in that circuit? So I think it's a promising area. It is happening. I think actually it's been helped by the fact that we've had advances in these other, maybe less elegant forms of treatment, of being able to just give a drug that increases dopamine or activate a network. I think we're better off to understand how to make stem cells work by having seen how successful, or at least partially successful, these other these other treatment methods have been. Yep.
There's another question here I like, because I believe that many of the diseases, or at least some of the major diseases we deal with, are predictable and preventable, or at least identifiable in their pre-symptomatic phase before people develop symptoms. Because, I don't know that a lot of people are aware that Alzheimer's disease actually begins in the brain 15, 20, 25 years before symptoms occur. Same thing with Parkinson's disease, maybe a decade or more. So there's a question here.
Same thing with Lou Gehrig's disease with ALS.
Exactly. So the question is, what biomarkers have been found? In this case, they're asking about movement disorders, but do you want to talk a little bit about biomarkers and the detection of prodromal or pre-symptomatic disease?
Yeah, I think so. There's that one slide I showed you of the PET scan with the alts in patients with Alzheimer's, where you can see parts of the brain light up very early, and we can use different ligands for these abnormal proteins that are accumulating and maybe see them earlier. That's one way. However, other really interesting new ideas include looking at markers that are in your blood. So you can get a scan and you might have an accumulation of the protein. You may not yet even have any brain shrinkage on MRI, but you may be beginning not even to have many symptoms. So how can we identify people really early so that we all believe that adding a treatment early is going to be the best protection? And also we know that behavioral changes like exercise, good diet, get rid of diabetes, get rid of hypertension, get rid of any of these metabolic abnormalities, exercise, sleep well, are all things that you could do if you realized that you were a person prone to this type of disease. But what's really interesting, and this is, actually, I know that one of the Lewy body dementia project that is being funded by ABF is going to be looking at this. There are little messages in your blood that may actually be coming leached out of the brain and are sitting around your blood, and we previously just ignored them. There are these little things called exosomes, and you can actually analyze the exosomes. So if you think of your brain, actually every night while you're sleeping, hopefully in deep sleep, there's sort of a process of like the little garbage trucks go around, take the refuse out, and out it comes through a system called the lymphatic system. And it gets sent out, and then it goes into your blood and out your kidneys or liver, as refuse. These packages of the garbage bags, if you will, they're coming out of the brain cells, have messages, and we're being able to track them. It's sort of like, you can't go anywhere because your iPhone knows where you are now. You can track which cells these have come from and see packages that are come from brain cells, and we can analyze them now and see that they have an abnormal signal in them, and that this person is in a more severe stage of the disease than they were a year ago, or they've been cured, or we'll be able to make those kinds of observations to help people understand where they are. So this is a very exciting area, this exosomal signaling. It can probably be used in the TBI field is using it actually. On the field. The idea is you could get a blood sample and see, oh my gosh, this person is these proteins are already in these vesicles, the refuse from the brain, if you will, is already sending a signal that there's something really bad going on up there, even though we don't see anything on the scan. So we're getting to that level of that level of resolution, and it's not just movement disorders, Parkinson's, Lewy body, Alzheimer's, and TBI. This could be used in epilepsy, in migraine even. We don't know yet, but this is, again, this idea that you can apply the same kind of method to multiple diseases is going to be funded by groups like us, like ABF.
Yeah. I mean, this biomarker research is, it's the future. And like you said, there's imaging biomarkers, there's fluid, there's tissue biomarkers. One of my colleagues has found that Lewy bodies can hang out in the salivary gland. So you biopsy the salivary gland, and there you go. You see the Lewy body that's characteristic of Parkinson's disease. And then there are just sometimes really simple biomarkers, like, for example, and ones that we don't pay enough attention to, like olfaction. You talked about the eyes being a window into the brain. Well, the nose is a window into the brain as well. And we don't do this rigorously enough in practice. But if you saw someone declining in with regard to their sense of smell and maybe moving around during REM sleep. Okay, well, we know that 10 years down the road, maybe that person might well develop Parkinson's disease or Lewy body dementia. So sometimes there are simple clinical things, clinical biomarkers or clinical markers that can predict the onset of disease. There's a question here, a good question actually, which I'm going to take from Valerie Pace, and that is, how does the American Brain Foundation collaborate with other disease-specific groups like the Michael J. Fox Foundation, for example, and organizations like that? And how do we build that collaborative model across interest groups? Frances, maybe I'll ask you to answer that, and then I'll ask our executive director to maybe weigh in on that.
Yeah, I will defer to Jane and you, but I know that we have in particular, so one of the most common and easy ways to have these collaborations is around the training of young investigators to help them develop this kind of mindset of being more open-minded outside of their single silo. And I know that ABF has one of the big, in addition to funding the research, it's also funding the training of next generation scientists that has to happen. And we're trying to change the mindset, I think, to make people more open to thinking across diseases. So the ABF has partnered with the AAN, it's partnered with the American Epilepsy Society to create fellowship programs for young investigators to get the start on their career so they can do all these amazing things that I've just shown you. I know that in addition, we are looking at specific sort of general areas like neuroinflammation. I talked a lot about inflammatory cells, for instance, and, thinking about like, okay, maybe we should get 4 or 5 different organizations to come together that each of them, I've said, it's involved in epilepsy, it's involved in stroke, it's involved in Parkinson's disease. So we could get these different organizations together under the umbrella of the ABF, or with the, using ABF as sort of the hub with those spokes to push a research agenda. So those are some areas that I know, and Jane has much more to say.
Yeah, you can expand on that, Jane. That's a great segue into the Neuroinflammation Initiative and how you've coalesced a group around that, and maybe talk a little bit about the Lewy Body Dementia Fund and the organizations that have been involved there as well.
Sure. Yeah, we have a research advisory committee of some of the most prominent neurologists in the United States. And we have that group looking specifically at what are some of the cross-cutting mechanisms that cut across many brain diseases, that if we were to create a research initiative, we could have an impact for multiple diseases at the same time. So one of those is neuroinflammation, as Dr. Jensen has mentioned. And we've been able this year to bring together people from the ALS Association, the Epilepsy Society, the Alzheimer's Association, the Multiple Sclerosis Society, the Peripheral Neuropathy Society, and the list goes on, to literally sit together for the first time in one room, one Zoom room, to talk about doing research together, and all looking at this neuroinflammation issue and seeing that, as one of our participants said, we can find a focus here that will lift all boats. So that's what we're doing with neuroinflammation. With Lewy body dementia, we're looking for a biomarker that will allow people to diagnose Lewy body dementia earlier than we can now. Right now, you can only actually diagnose it for sure in autopsy, which isn't very helpful to the people that have it and their caregivers. And so we're looking for a way to diagnose that. But as it turns out, our biggest supporters in this research are the Alzheimer's Association and the Michael J. Fox Foundation. Why? Because Lewy bodies are involved in those diseases as well. And they understand cure one, cure many, that if we work together on mechanisms that are cross-cutting and symptoms, et cetera, that are cross-cutting, we can make breakthroughs for everyone. So, and then I'll just lastly say that we have partnerships with about 17 or 18 national single brain disease organizations. So from the Alzheimer's Association to the ALS Association to the Parkinson's Foundation, and on and on and on. And so we're, as the American Brain Foundation, more and more finding ourselves and liking finding ourselves in the role of a broker that can bring the different groups together around different shared mechanisms. So it's very exciting, and our whole, we're working on strategic planning right now, and one of our whole issues is how to be an outstanding partner. We want to do that with all the different entities that are out there, including pharmaceutical companies, academia, you know, to able to come together around these issues.
Thank you, Jane. Perfect. And including two organizations that are outside the field of neurology, right? So we're breaking down silos within neurology and outside of neurology. Frances, I love the way you broke down that global burden of diseases study, where you basically lumped many diseases that are truly neurological, making it very clear that it's clearly at the top. And so one of the things you said is, one of the things you lumped or you tied with traditional neurological diseases are mental health disorders. And there's a question here, should they be bifurcated into two different categories, and what's the relationship there?
Okay, so look, my approach to brain is that it is, you know, composed, as I said, of a myriad of component parts, but they're rearranged in space and time to create different neurological and psychiatric diseases. So we see psychiatric diseases all the time inside neurological disorders, and vice versa. In fact, we share drugs. So a lot of the antipsychotic or depressive drugs are actually relatives of anti-epilepsy drugs. I mean, we know we're dealing with the same neurotransmitters, serotonin, glutamate, we have drugs that are being used in both settings. And what we're realizing is and some of our diseases like limbic encephalitis start out like a psychiatric disorder for a few days or weeks and then turn into a neurological disorder. Of course, they're related. I think neurology is ahead of psychiatry. I mean, clinical neuroscience is like where in its evolution, where sort of cancer oncology, that field was 10 to 15 years ago. We're catching up fast. We're moving fast. I think neurology, because we have more obvious diseases that kind of are more straightforward and might involve certain parts of the brain that you can see on an MRI, it's churning ahead. But a lot of what we are doing applies right back to psychiatry. Psychiatry is essentially, in my mind, subtle neurology. it's the not obviously visible, but it's might be physiological differences that we see in neurology, that in neurological disorders, but they're in different places and not as maybe extreme in some cases. I think we can connect the two fields. We've been talking about this at board meetings, et cetera, a lot, like it's really one field. I do think we have an advantage in neurology right now over psychiatry in that we have so many tools. Psychiatry is going to get there, but it's behind in terms of the molecular biology. It's getting there. Genetics, they're finding all kinds of genes are dysregulated in psychiatric diseases, just like we do. And they're starting to use functional imaging, like we saw, to show how brain areas are connected the wrong way. And they look the same on scan, but they're not talking to each other normally. And that you can capture that with functional imaging. We're seeing that. And so I think the fields are going to be coming closer and closer. And we won't be able to ignore, psychiatry and wall it off. It's in us and we're in them. There's no question. And we're talking about training people across those barriers a lot too, especially on the clinical side. I did just want to mention, I see a couple of people have been talking about frontotemporal dementia. Yeah. I have to mention it because there's an example of a disease that is a disease that is so connected to other diseases. So TDP-43 is one of the proteins that's accumulated in frontotemporal dementia, which has a different pattern, different parts of it has a different course than Alzheimer's, can be affecting the more emotional parts of your brain first and in your judgment, so that your memory may be good, but you're really not interacting with other people in a productive way, and you're anxious and emotional and very, it's affecting, the degeneration is happening in those parts of the brain as opposed to just purely, say, the memory part of your brain. And there are other kinds of dementia that affect other areas as well. So frontotemporal dementia is differentiated from Alzheimer's in that TDP-43 is one of the proteins that is seen, but it can be seen in Alzheimer's too. It's just more there in frontotemporal, and it's also been seen in Lou Gehrig's disease, in ALS.
Mhm.
So TDP-43, again, is one of these on this list of proteins that can connect a number of different diseases, and we can study it in these different settings to get a better understanding. And maybe one day what we learn from TDP-43 and frontotemporal dementia might actually help people who have a form of ALS that's caused by this.
Yeah.
There are other proteins like progranulin that are pretty, more seen in that disease. And again, we're showing that there's like 12 to 15 types of dementia. It's not one dementia anymore. And frontotemporal dementia is a perfect example of like how we've been able to fine tune. These people are different. They look different, they act different. And now we have data on them about their molecular biology and their genes and their brain function that are different too, because we can apply these new tools.
Yep.
We know it's connected. We know that some of these elements are connected to other diseases to help us study the mechanism.
Yep. Frances, Roxanne asks about how to get studies to look at families and what about genomics, and not just in clinical trials either, or in studies, but also in clinical practice. I mean, you were at Harvard and they had the first preventive genomics clinic in the world come out of Harvard. And, although we have 20,000 genes, apparently there's like 75 actionable genes. So what's the role of genomics in clinical practice and in clinical trials?
We have thousands of genes now identified as part of neuropsychiatric disorders. In epilepsy alone, for instance, there's probably 250 or 300 known genes that are, attached to different syndromes. Look at autism, look at all these different named conditions that are now because of a genetic mutation. So this has come out of population health, genomics, genome-wide association studies, and now much more specific genomics. And what's amazing is it's helped by social media. People say, well, my aunt had this, and I got this kid or this cousin that doesn't look right either, and people are connecting. We're seeing patient groups that are identifying. I work with CDKL5, which is a form of autism, neurodevelopmental disorder. It's one of the many things I work on. It has some epilepsy. It's an early life, pretty severe epilepsy, and can have a severe intellectual disability early in life. And 20 years ago, nobody even knew there was a CDKL5 gene. It hadn't been even named. I made a diagnosis back when it was first on an Athena panel of, a rare disorder. And we made the diagnosis, on a patient that I've become very close with, that family and that organization. And and since that time though, because so many people are being told what gene they have, they go online, they find an association. There's now several CDKL5 associations for patients, and the patients are organizing themselves and asking questions. And the patient here is talking about frontotemporal dementia. There's a frontotemporal dementia group with specific genes that, for people with TDP-43, gene problems or progranulin. And what we're getting is this great kind of partnership now with patient groups of what used to be thought of like rare diseases that you would come across. You're seeing thousands of cases when you thought you might only happen upon a handful in your lifetime, because the people are presenting themselves. And it's this great interaction between patient populations and scientists now.
Yeah. Well, unfortunately, we're at the top of the hour, and obviously we could go on, and there's many questions I didn't get to, and I apologize for that. But that just means, Frances, you're going to have to come back for an encore presentation.
Delighted to.
and answer some of these questions and more. So Frances, thank you so much for doing this. It was a fantastic presentation. You got me jazzed. I'm sure that's true of everybody on this call. Thank you all for being here tonight. And some of the upcoming events you'll see here, there's talk about, Frances talked about deep brain stimulation. So we're going to have a session on deep brain stimulation. You're going to meet some of these young researchers that Frances talked about that we fund, remarkable young clinician scientists. Managing Parkinson's disease and multiple sclerosis, Frances talked about that tonight. We're going to go take a deeper dive into that. And navigating migraine, some of the new treatments. I mean, 8 new drugs, though, just over the past 2.5 years. It's remarkable after 2.5 decades of desert. So it's lots of excitement there too. So please tune in for those. And thanks again for your generosity in supporting the research that we fund here at the Foundation, American Brain Foundation. And we'll see you next time, Frances. Thank you again.